Some people think from movies and television that when you have a clone, the clone looks exactly like you, but that's very unlikely. Genetic engineering is the manipulation of genetic material in a cell or an organism in order to produce desired characteristics and eliminate unwanted ones. GE includes a range of different techniques with many different uses and can be applied to plants, animals, and humans. For example, the genetic modification is a form of GE that involves manipulating the cells of plants such as maize to increase the yields. Cloning a human is certainly the ultimate challenge for genetic engineers. Cloning is not limited to this goal. Cloning can be and is done on a much smaller scale and could involve no more than just the cloning of a single cell. Therefore, cloning can be divided into two types. In order to make a clone of someone, one needs a living cell and human egg. The nucleus of the egg, which contains the DNA, is removed and placed with the nucleus from the cell and person animal to be cloned. In therapeutic cloning, an embryo is created in the same way as reproductive cloning, but is not implanted into the womb of a woman. Instead, stem cells are extracted after the embryo, such as to divide in the first 14 specific type of tissue or organ that is needed and has the advantage of being genetically identical to the patient who is donating it, eliminating the problem of organ or tissue rejection. Replacing organs and other tissues, such as new skin for burn victims, brain cells for those with brain damage, spinal rod cells for the paralyzed, and the complete new organs, health, liver, kidney, and lungs. Pigs are also being genetically modified to make their organs more compatible with humans by removing the gene that causes rejection. People could have their appearance change, cosmetic surgery, using their own clone tissue, and accident victims and amputees could also benefit from this tissue regeneration. Reversal of aging process. Once more is understood about the role that our genes play in the aging process. However, some of the above uses carry with them some serious ethical implications. Human cloning provides couples and individuals who are unable to have children with another potential option. Donor people, clone people could be created to provide a source of transplant material. Saving endangered species by boosting their numbers through creating clones. However, since clones are almost genetically identical, the genetic diversity of the species will not increase. Low success rate. Dolly the sheep was successfully cloned. It took 276 unsuccessful attempts before it worked. Similar work on mice and mammals has also produced the same statistics to date. The success rate on animals. Embryonic stem cells are unstable and difficult to control. They have a tendency to uncontrollably divide, leading to tumors or cancer. Clones of animals are larger than average at birth, which can be risky for the mother. The age of the clone is calculated by taking its birth age and then adding the age of the original from which it was cloned. Although Dolly was born in 1996, she originates from the udder of a 6-year-old eel, and so her total genetic age is almost 13. If applied to humans, the current method of cloning would use a vast number of human eggs. To provide these eggs, women would have to become egg factories, and harvesting them is painful and dangerous. If adult stem cells were used, then human eggs would not be required, as cells could be obtained from the patient without harming them. Since by nature a clone is a copy of another person, there would be no unique genetic combinations introduced into the human gene pool if human cloning was undertaken on a large scale. Therefore, if a contagious disease struck from which there was no cure, all clones would be wiped out. In gene therapy, where a healthy gene can be used to replace a defective gene, viruses are usually used to insert the gene into the person's cells. The virus infects the healthy DNA into the cells and the genetic defect is corrected. However, this is not always successful as the virus cannot be controlled and has triggered leukemia in recent clinical trial. And although the Human Genome Project has mapped out where the different genes are, a lot more information is needed for their functions. In some cases, a single gene may have more than one function, and in others, several genes can cause a genetic disease. In the US, there is no public funding for research on embryonic stem cells. Therefore, although there's no law against therapeutic cloning in the US, there's almost no public research happening due to the lack of money and access to the embryonic material. Simultaneously, an announcement was recently made stating that the US public funds about $1.4 million will be provided for the studies using adult stem cells instead of embryonic stem cells. The Chinese government is funding an extensive research and drawing back many Chinese scientists from overseas to undertake work they would not be allowed to do elsewhere. With easy access and no limits on obtaining the embryonic material they require, it is expected that the Chinese scientists will race ahead of the rest of the world in the therapeutic cloning technology. South Africa, currently the Human Tissue Act prohibits both therapeutic and productive cloning of humans. This situation will change when Chapter 6 and 8 of the National Health Act are promulgated, and therapeutic cloning will then be allowed under strict conditions requiring ministerial permission, but reproductive cloning on humans will remain strictly prohibited. In the UK, there are clear rules banning productive cloning, but scientists in both the UK and Israel are allowed to generate new embryonic cell lines for therapeutic research. That's it on genetic engineering and cloning. And remember, clones don't always look exactly like you.